we are going to demonstrate how you design a RFLP experiment. Remember what is RFLP? RFLP is restriction fragment length polymorphism. And you already have read about restriction endonucleases, which are bacterial enzymes, which are restricted to particular sequences of viral DNA. If those sequences are there in the viral DNA, the restriction enzyme will digest the viral DNA and break it into smaller parts. Remember, restriction endonucleases will not digest methylated DNA sequences. So the bacterial DNA is actually methylated. That's how the enzyme differentiates between the bacterial and viral DNA. So all these restriction endonucleases are commercially available and they are used for diagnostic purposes. So we'll design a particular experiment where we will use the primer sequence that we have designed previously for HBB gene, and we'll try to find out if any particular restriction enzyme is there that can digest that sequence and break into smaller parts that can be differentiated by a gel electrophoresis. For example, agarose gel electrophoresis. And in that particular sequence, if there is any mutation, then we will find able to identify that mutation based on whether the DNA is digested by the enzyme or not. So how do we do it? We go to a particular website called NEB Cutter. We go to NEB Cutter website and okay. So you paste the sequence that you had amplified by the in silico PCR. Paste the sequence and submit. Okay. So you see that there are many enzymes that can cut it. So we want to pick a particular enzyme for custom digest. So these are the enzymes which are available, which have got a unique sequence. We pick any one of that. So we will pick only those enzymes which cuts at one place because otherwise it becomes difficult. So let's pick this particular enzyme BAM-H1. So what is BAM? BAM is the name of the enzyme, the bacteria. B is the genus. M is the species. H is the strain and I is the number. So if you search for BAM-H1, you'll know the name of the bacteria, which is this. So B is for bacillus and AM is amyloliquefaciens. That is the full form of BAM. And H is the strain and I is the number. Okay. To select BAM H1 and then digest. So after digestion, how will be the products? We see the gel picture. We want to make it a little bit more percentage of agarose, 1% agarose, and we put a marker under display DNA level. Okay. So this is 100 base pair, this is 200, 300, 400, 500, 570, like that. Okay, 600. So this is one fragment that is coming 91, another is 500. So the sequence where the BAM H1 had digested the DNA, you zoom in, further zoom in. Okay. So this is the sequence where it is digesting. If there is any mutation in this particular sequence, then this digestion will not occur. So then we will say this is a mutation. And if the digestion doesn't happen at all, so the total product is 591 somewhere here, then you say that this is a homozygous mutant allele that is there. If three bands are there, that is one band is cut, another was not cut, then you got it a heterozygous. And if similar to that, two bands are there, then you call it normal homozygous. Okay, this is how you design a RFLP experiment.